this is JJ, and I will be talking about determining how S100A9 activates TLR4 using evolutionary and biochemical approach. The immune system activates inflammation in response to both foreign pathogens and internal damage. Dysregulated inflammation can lead to many chronic diseases such as cancer. Both LPS and S100A9 are able to trigger such immune response. LPS is found on gram-negative bacterial cell membrane that goes into the system with the membrane and is able to activate TLR4, which is a pro-inflammatory receptor, and thus activates NF-kappa B signaling cascade and activates inflammation, trying to kill the bacteria before it's able to harm the host. Interestingly, S100A9, which is a protein expressed in the immune cells in the host, has been found in high concentration in inflamed tissue of many of these chronic diseases. It turns out that S100A9 strongly activates TLR4 as well and causes such pathological inflammation. Therefore, understanding of how S100A9 interacts with TLR4 would be useful to create therapeutics to treat such diseases. And that's why I'm asking how and when did S100A9 evolve a pro-inflammatory response with TLR4. Based on the previous existing data and research in the lovely Hans lab, we know that chicken S100A9 activates TLR4 much lower than opossum S100A9 does, lower than mouse S100A9 does, lower than human S100A9 does. And that sounds like a trend because chicken diverged in the evolutionary tree of life earlier than opossum did, than mouse did, than human did. So, that makes me wonder if there's a connection between S100A9 evolution and the species evolution, which, based on the species tree of life, that chicken diverged before the ancestor of all mammals. So I am hypothesizing that S100A9 evolved to more strongly activate TLR4 in the ancestor of marsupial mammals, and I'll be testing my hypothesis by using characterizing more than a mammalian S100A9s that diverged more distantly from humans, such as koala, platypus, and echidna, using recombinant protein expression and purifying S100A9 protein from E. coli, followed by the transfection assay in human embryoid kidney cells. And I'll be measuring NF-kappa B output, because that's the signal sent to activate inflammation. And in my expected result that opossum and koala are very closely related, so they should have close ability to activate TLR4. Platypus and echidna are very closely related, and they diverged after the ancestor of all mammals, but before the ancestor of marsupial mammals. So they should be close to each other, but much lower than koala and opossum, however higher than chicken. And comparing opossum TLR4 and human TLR4, the three new species that I'm testing should have higher activation towards opossum TLR4 than human, because they are more closely related to opossum than they are to human. And I have some these are my interpretations. And if my data actually appear to match my expectation, that will support my hypothesis that S100A9 may actually be evolving alongside of species following the evolutionary tree of life. However, if it does not match my expectation, it will show that my hypothesis may not be correct, and we need further investigation and experiments in terms of exploring the evolutionary tree of S100A9. thank you so much, and thank you to everybody in the lovely Hams Lab, and thank you for your time and attention.